DNA transcription, DNA replication, and the regulation of gene expression all depends upon the recognition of DNA by proteins. We need to understand the structures of double-stranded base-paired helical DNA molecule on its own in order to see what possibilities it offers for the recognition of specific sequences by proteins. This is a physiological DNA, B form. Sugar phosphate backbones go anti-parallel 3 to 5 prime and 3 to 5 prime. Notice that the central axis of double helix goes through the middle of the base pairs, that the base pairs are perpendicular to the axis. There are two types of only base pairs, AT and GC. In addition, there is some highly dehydrated DNA form, that is A form, and that could be found in double-stranded RNA. And also, recent studies just shows that there are G type of DNA also possible in sequence-specific regions like CG repeating unit. Three types of DNA are shown here with top view and side view, each containing 22 nucleotide pairs. Top structures belongs to B form. You can see clearly major group and minor group. Center structure is A form. You can see some hole along the helical field, this one, and also the thick and short compared to the B form. C is G DNA. Has quite different structure. Helix is left-handed compared to the right-handed for the other two, and the sugar phosphate backbones just follow zigzag path. The shape of this helix is thin and elongated. It has a deep but quite narrow minor groove here, whereas the major groove is pushed to the surface so that it is no longer a groove at all. We must understand what causes major groove and minor groove in B form. In this hypothetical regular helix, the steps attached to the sugar phosphate backbones equally both sides, so that the two grooves, front and back, they almost identical for their shapes. However, in B forms, base pairs attached to the sugar phosphate backbones in a symmetric way, allowing wider the groove called major groove on top, and the narrow groove called minor groove at the bottom. In B, you can see AT and GC base pairs. Actually, the way base pair is connected to the sugar phosphate this way and this way. And again, you see a symmetric chemical bond. This slide shows a schematic diagram illustrating major and minor groove in A and B DNA. In A DNA, helix is shifted from the center of bases into the major group, bypassing the bases, and the base pairs are not perpendicular to this axis but are tilted between 13 and 19 degrees. In bDNA, because the helical axis runs through the center of each base pairs, and the base pairs are stacked nearly perpendicular to the helical axis, the major and the minor groups are of similar depths from surface, from here to here and from to here. The difference between them are just widths, axis of a certain surface, wider and narrow. Depths are almost identical. This table just summarize B and A DNA in comparison with the regular helix. The location of base pairs to the backbones slightly toward the minor group in B forms compared to the being prominently toward to minor group in A forms. Axis of double helix penetrate through center of base pairs in B forms, but major group in A forms. Angle of base pairs to the axis is almost perpendicular to in B forms, while a little bit tilted in A forms. Depths of two groups are almost identical in B forms, while one being very deep for major group in A forms. Let us now look at how specific sequences might in principle be recognized in bDNA. The only regions where the bases are available for interactions are at the floor of the groups. The edges of base pairs contains nitrogen and hydrogen atoms that can make hydrogen bonds to protein side chains. So GCAT has a hydrogen donor NH, and GA has N, and GCT have O, which could function as a hydrogen acceptor. In addition, T has methyl group, as you see here, this methyl group, and A has hydrogen. Okay, so this methyl and hydrogen also provide the binding site for the protein side chain. This kind of different interactions just denoted with the different colors as noted here. By using this color code, you can see this kind of pattern. Those W indicate wider, meaning major group side, and S indicate small, so that it could be correspond to the minor group. By using color code on top of this slide, you can see color picture of tetranucleotide pairs G, A, C, T from the side of major group and minor group. First of all, each major group has 4 binding sites, whereas only 3 binding sites in minor group. And also you see the patterns are different for each individual base CACT. However, minor group just giving identical patterns for G, C, and A and T. This slide shows sequence-specific recognition sites in the major group of DNA for 3 restriction enzymes, EcoR1, DEL1, and SMA1. DNA sequences that are recognized by these enzymes are represented by the color code. You can see clearly 3 sequences are different. These patterns are quite different, and each can be uniquely recognized by specific protein DNA interactions. In sum, major group provides much better available site for the sequence-specific recognition by protein, because simply it could provide wider space and more specific patterns to the protein.